Good evening. It's Dr. Pierpont here again, and this is our third session of our, yes, our fourth session of live Q&A. Tonight we're going to be talking about meiosis and mitosis. Um, this is a process by which we create new cells, and we're going to start with mitosis. Mitosis is for the purpose of growth, repair, and replace. Those three things: growing an organism from a seedling to a flower, a baby to an adult human, um, repairing. When you get a cut, you have to repair the cut, and cells are produced for that. And then to replace. Cells do die eventually, and when they die, they have to be replaced. Some of them are very important cells, like in your heart and your liver and things like that. Brain cells never get replaced. They get grown and they get put in place, but once you lose a brain cell, that's it. That's why drugs are bad for you, because you lose brain cells. They never grow back and repair. So in mitosis, you have growth, replace, and repair. In meiosis, you have a different format of working with the cell, and we're going to examine the differences very closely tonight. So let me first off start by saying, you guys need to show up for these meetings. This is for your benefit, and you can ask questions. Maybe you don't have any questions, but it sure helps to get a heads up on what's expected for you to do in your next assignment. So think about that and show up if you can. Otherwise, you have to write a paper and a summary about what it is that I talk about. So going back to mitosis, in the cell itself is a complete set of DNA. And this complete set of DNA is possible because we have a nuclear membrane. We talked about that last week. That nuclear membrane in eukaryotic cells makes it possible to have all of this DNA inside the cell. And it would be possible for me to clone you if we wanted to. We could clone you, and it wouldn't be very hard to do. We do it all the time. We clone pigs, we clone sheep, we clone tomatoes constantly. We've been doing this a long time. So it's easy to manipulate the DNA because of the very simple way that DNA copies itself. So you have a DNA strand, right? And I'm going to use my fingers all twisted up here. This is the double helix. This is what you're used to seeing when you look at DNA. That ladder that's been twisted, that's called a double helix. And this double helix is twisted on itself, and it makes a copy, an exact copy of the opposite of itself, like looking in a mirror. If you've ever looked in a mirror and raised your left hand, it looks like you're raising your right hand. All right, so we're making an exact copy of the opposite. Now, in DNA, you've only got 4 chemicals, 4 amino acids: adenine, thymine, cytosine, and guanine. A, T, C, G. All right, so A goes with T. It only goes with T. C and G only go together. You never get C and T together. You never get G and A together. A and T makes the word at, so you know that you're on the right track. If you can form the word at, then the remaining 2, C and G, go together. So in this mirror copy, the A will make a T, the T will make an A, the C will make a G, and the G will make a C. And it makes a copy of itself. When it does, it then splits away from itself and goes to opposite ends of the cell. These opposite ends of the cell then form the mirror image of their opposite. So A to T, T to A, C to G, G to C, T to A to T, G to C, C to G. All right, so they make a mirror image of themselves. They pull to the ends of the cell, the opposite ends of the cell. They pull like this. They actually at one point form the letter A, and that's called anaphase. And we'll talk about the different phases of mitosis in a second. And then the whole cell splits apart, making two daughter cells. We call them daughter cells because they're identical to each other. You could not tell these two cells apart. They're identical. All right, so the steps of mitosis are interphase or S phase 1. Okay, so during interphase, this is when all the copying is happening. We used to think that the cells slept during interphase, but it's actually the busiest time because it's making copies of all of that DNA. And if you stretched your DNA out in every cell, would go past 36 meters. In every single cell of your body, of which you have trillions. You could stretch this and go to the moon and back a couple of times if you stretched all of your DNA out into one long string. So you've got you're very informationally packed. So in this interphase, all of the copy is being made. Then we have prophase. This is when the two sets of chromosomes on the DNA line up. Okay, prophase, metaphase, they're all in the middle, nice and tightly packed. Prophase also has the spindle fibers being formed. These are going to be useful for pulling this apart. So prophase, we've got spindle fibers. Metaphase, we've got everything lining up in the middle. Meta means middle, so this is the middle section. Then we have the anaphase, where the cell pulls the two sets of DNA apart, anaphase. And finally, we have telophase, which is when the cell pinches itself in the middle and makes two daughter cells. So you can remember this by IPMET, okay? Interphase, prophase, metaphase, anaphase, telophase. All right, so IPMET is the way that cells actually make a new copy of themselves, and they are identical. You could not tell them apart. So this is the first step in grow, replace, and repair. Now, this works great because, like in humans, you've got 46 chromosomes here, you got 46 chromosomes here. You could clone a human out of that, but you can't make a new human out of that. And here's what I mean. If you clone a human, you're just making a copy of a copy, okay? But if you want a brand new human, a baby, or a brand new flower, not a copy of a clone of a flower, but you want seeds, okay? In order to make seeds, you have to go through meiosis. Meiosis is almost the same as mitosis, but it goes through the division process 2 times instead of just 1. Interphase, okay, interphase, prophase, metaphase, anaphase, telophase, all right, 2 new cells. Then in meiosis, you go through all of those steps. Then you go through interphase again, but you don't copy the gene. You don't copy the DNA. Instead, you take the DNA and you cross it over a little bit. And this crossing over shuffles the genes that are in the DNA. Then you go through, after you've done that, now remember, you're not copying the DNA. What you have then is prophase, spindle fibers again, metaphase, All right, everything's lined up in these two separate cells, not together, but in these two separate cells. And then they split apart, each one having exactly 1/2 of the 46 chromosomes that make up a human. Each of these little cells have 23, 23, 23, 23. That's why it's called 23andMe. Okay? So 23 chromosomes in each of these cells. These are what we call gametes. Gametes are egg or sperm, and this is how they are made. In flowers, same thing happens. In animals, in bugs, anything that reproduces sexually goes through this meiosis process. And this meiosis process, then from that, you have 23 chromosomes in each one of the cells, okay, for humans. Now, for bananas, you have 1,000 or more. For dogs or chimpanzees, you have 21 or 19, depending on the species of the animal. So you've got these halfling, as it were, cells. Now, how does it work? In fertilization, you're going to have half of these 23 for humans in an egg meet the 23 in humans in the sperm, and you end up with fertilization. This fertilization then makes 46. Okay, so 23 and 23 make 46. Well, humans are supposed to have 46 chromosomes. So in these chromosome matchups here, we end up getting the full amount of necessary chromosomes to make that organism. Now, things can happen along the way. Sometimes a chromosome will drop out. Let's go to horses and donkeys. If you take a female horse and a male donkey and you mate them together, you end up with a mule. Now, a mule has horse and donkey, horse and donkey, horse and donkey. Uh-oh, you're missing one. There is no extra chromosome to match up with this chromosome. It's missing. The donkey is missing one of the chromosomes that the horse has. This is what makes the mule. And a mule is always sterile. They cannot have baby mules. Mixing up certain profiles of genes can make for disasters. If you take alpacas, for example, fluffy alpacas, beautiful alpacas, and you have a female blue-eyed alpaca, all right? And then you have a male white blue-eyed alpaca. If you mate those two white blue-eyed animals together, the baby is born with a defect of having no face at all. No eyes, no nose, no mouth, just not there. And considering that you have 365 days of gestation with alpacas, and alpacas are still fairly expensive to buy, that can be a horrible thing. So the first thing we do when we have a male blue-eyed alpaca is we geld him. And that way he can't breed with the female and end up having this horrible thing happen with the babies. So over time, genetics have become very interesting to the scientific community, and we now play with them frequently. Some of this is good and some of it is bad. Some of it's very good. We have figured out that we can take a cell and we can print it and we can make skin. Now, this is really good because if you're a burn victim, we can take a little pinch of your skin and we can grow sheets of skin to put onto you so that your body will have skin on it again and it won't reject it because it's your skin. We'll get more into that next week when we talk about cloning. or week 5 when we talk about cloning. And this is week 3, not week 4. Sorry, my bad. Got tripped up on that a little bit. So we're on week 3. So anyway, these two factors, meiosis and mitosis, are what keep the body running. Every 7 years, you replace all of your cells, given enough time. You have trillions and trillions of cells in your body. But if you take a look at a baby, a 7-year-old, a 14-year-old, a 21-year-old, and so forth, you can see the evidence of how many times the body has taken every cell and made a new copy. You make new copies all the time. Cells die. Lifespan of a cell can be about 16 to 21 days when you're in your teens. For me, being older, my cells are dying faster than I can replace them, which means that I am approaching what will be death eventually. Because when you stop making your cells, like your hair here, turns gray, no longer it makes color, so you end up with gray hair, and your body begins to break down, and you're not replacing like you should. Your cells are no longer making copies. So that's why we want to stay healthy. We want to eat our good vegetables. We want to take our vitamin D. We want to take our B12s, and we want to do those things that make us healthy again. So this is all that I have for you for a lesson tonight, and I hope that you write up your summary, study mitosis and meiosis, and write your summary up about kind of contrast the two um, figures there, the two examples of cellular reproduction, and that would make a really good paper. All right, I will see you then. Hopefully, I'll see somebody. I'm kind of alone out here, and I'm, tear, waiting for you. So, hopefully, you can join me next week, and we'll talk some more about how not to have ugly babies. That's what we're going to talk about next week. So, I'll see you then. Bye bye.